Hey folks, and welcome to another edition of Zog Science. Today we're going to be taking a look at the last piece of the central dogma, which is translation. So just a little reminder of where we've been. We've got a gene, right, which is made up of DNA. That DNA can be replicated to form some more DNA. However, that gene has been read and turned into a piece of pre-mRNA. The pre-mRNA was then modified to only contain specific information necessary for the protein and is now going to be sent out from the nucleus to either the rough endoplasmic reticulum or the cytosol, where it is going to meet up with a ribosome. And ribosomes are the factories of cells. They produce proteins, and they are made up of rRNA, which is ribosomal RNA, which makes sense, and proteins, and it is assembled in the nucleolus, only in eukaryotes, obviously, because the nucleolus is located in the nucleus, and it consists of a large and a small subunit. You can kind of see what that looks like over here. This is more the molecular structure. This is just kind of a diagram, and you can see the small subunit. The mRNA will bind to the small subunit first, and then the large subunit will kind of attach onto there. We have three different sites. The A site is where the tRNAs will kind of come in. The P site is where the amino acid from the tRNA is added onto the growing polypeptide chain, and then we have the E, or the exit site. So this is translation that we're talking about. We're going to be taking mRNA and we're going to be turning it into a protein, and we're going to use a triplet code, which is the codons, to place amino acids in the correct sequence. So it is about the specific sequence of the nucleotides found on the mRNA that is going to lead directly to the protein being formed. The different players in this play are mRNA, which is going to carry the genetic information, although it's in a slightly different form from the information what it was in the DNA, right? So it's been modified, so it's a little different now. We've got the ribosomes, which help facilitate the process of turning the mRNA information into a protein, and then we've got the tRNA, which is going to transport amino acids to the ribosome. So let's take a look at the tRNA. This is the structure. It is, again, just a piece of RNA. However, there are hydrogen bonds that are going to be taking place between different bases, and that's going to help kind of hold it together and give it its specific structure. We have an anticodon, which is going to pair with a codon in order to help align the amino acids. The amino acids will then attach onto the opposite end of that piece of tRNA. Now, to kind of get started with, we're going to have a start codon. So first, the mRNA binds to the small subunit. The mRNA is then read until you get to the first sequence of three letters, that is AUG, and it will just read off one at a time until it gets to that first sequence of AUG, and that codes for methionine. Methionine always goes at the start of a protein. Every single protein has methionine at the start. Now, it can have methionine later on. That's not a problem, but it always has methionine at the start. Once that tRNA with the methionine is in place, the large subunit will then come and attach around it and facilitate the rest of the process. So the next part of the process is called elongation. We've seen elongation before when we've been dealing with the production of both mRNA and the production of DNA. So first things first, the next codon will pair up with the anticodon of a tRNA, which will bring in the correct amino acid. Then the amino acid will be added on to the growing polypeptide chain. So the peptide will come off of the initial tRNA and then added on to the next one. You can see that kind of here. And then they kind of are pulled through. So the piece of RNA is pulled through. When the tRNA gets to the exit site, it gets kicked out. And now this cycle continues. We just keep on adding on three letters at a time, three nucleotides at a time, one codon at a time, each amino acid. Once we get to the end, we have stop codons. There are three stop codons, and they code for a release factor. And when the release factor comes in there, as it gets pulled through, the polypeptide is released from the last piece of tRNA, and then finally the small and large subunits break apart from one another. This does take energy in order for this to occur. Energy in this process comes from GTP instead of ATP. For us, that kind of details the process.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
All right. So when we're going through the process of translation, we can actually have multiple ribosomes attached to a single piece of mRNA. And that allows us to create a lot of different copies of the polypeptide at once. And this happens in both eukaryotes and in prokaryotes. Now, prokaryotes, because they do not have a nucleus, transcription and translation can actually happen concurrently, right? They can be happening at the same time. Here's how. The RNA polymerase produces the mRNA. As the mRNA is being produced, ribosomes will attach to it and begin to produce the polypeptide. So the processes can be occurring at the exact same time, which I think is a really cool thing. It just seems like a really cool adaptation, right? It makes sense. You don't have to wait for the whole piece of mRNA to be produced in order to get that translation process started. So pretty cool. Mutations. All right, we can have mutations. Mutations are changes in the DNA code that will lead to a different protein. Now, they don't always necessarily have a negative effect, and we're going to talk about some examples why. So before we do that, I got another quick video for you about sickle cell and the mutation that causes sickle cell.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under low oxygen conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in a position where glutamic acid should be. The valine makes the hemoglobin molecules stick together when oxygen tension is low, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
One of the things I really like about those animations is that they kind of show the molecules all moving around, right? And that makes sense because they all have kinetic motion. They all have temperature. So that's one of the really cool things I like about those videos, the kind of depth of the detail. So now I did mention that some mutations don't always have an effect. We call those silent mutations. And the reason why is because we can have multiple codons that code for a single amino acid. If you take a look at a codon chart, usually there are, about 4, sometimes there's less, sometimes there's more, but usually around 3 or 4 codons that will code for a single amino acid. And that allows us to have some mutations that can occur that won't always have an impact on the protein, which means that the mutation has occurred, no phenotypic difference, which is a good thing. Now we can have a missense notation, a missense mutation, an example of which is a substitution where one letter is substituted for another, and that leads to a single amino acid difference. So we have now a new protein that has been produced. We also have nonsense mutations. We can have a substitution that will result in an early stop codon. And so essentially no protein is produced. We call that a nonsense mutation. We can also have a frame shift mutation. Here's where we either have one base that is either inserted or deleted. And what happens with that is that either what happens is that the frame shift gets moved over, right? The reading frame. So remember, the reading frame is every three nucleotides after the start codon. So when that reading frame gets shifted over, it can lead to either a nonsense mutation where we have no protein produced, or we can get a missense notation where the stop codon never gets placed in. Therefore, the protein just keeps on going on and on and on, and we end up with this huge protein that can't do anything. But those are caused by frame shift mutations where either an addition or a deletion of one base leads to lots and lots of problems downstream of that insertion or deletion. Now, we can have a frame shift mutation where we insert or delete 3 nucleotides. And this is going to be generally much less of a problem than the other frame shift mutations, for the reason being that we're going to take out an entire codon or delete an entire codon, either add or delete a whole codon. Therefore, we're only going to be affecting one amino acid. So not as big of a problem. So just to kind of summarize everything we've been talking about the central dogma, which is taking DNA, turning it into RNA, turning it into a protein. Now we can also have DNA replication occur where DNA is turned back into DNA. That's obviously very important for proliferation of the information from generation to generation. And just kind of a sentence to kind of sum all this up. The genetic code is punctuated, unambiguous, and redundant. It's punctuated. There are clear stops and starts to the information, right? There are genes that have starts and stops to them. It is unambiguous. There is a specific set of instructions that codes for a specific protein. However, it is redundant. You can have several different codes that will lead to the same proteins being produced, right? So, or rather, I guess the same amino acids. So we can have different codons that lead to the same amino acids being produced. And remember, this is the molecular basis for life. If we don't have this, life cannot exist. So I hope you enjoyed it. Thanks for watching, and I'll see you next time.